So hi everyone, and welcome to the 18th TIA Centre seminar of the academic year. So my name is Adam Shepherd, and I'm a postdoc here at the TIA Centre. Those people online that are new to seminars, we aim to invite researchers from across the globe to present new and exciting work. And today I'm delighted to introduce Dr. Sebastian Klein. Sebastian Klein studied medicine at the University of Munster and spent a research year at Harvard Medical School. He then completed his residency at the Department of Pathology in Cologne and Munster. His scientific training included data analytics from unstructured and structured data, as well as translational cancer research with a focus on the tumor microenvironment. After moving to the Department of Hematology and Stem Cell Transplantation in Essen, where he built an analytical ecosystem for molecular assessment and reporting of hematological cancers. His primary interest shifted to translational lymphoma research and diagnostics. Personally, he enjoys endurance running, Swift virtual bike racing, and he's a father of 2 little kids. And the title of Sebastian's talk today is Predicting HPV Association Using Deep Learning and Regular H&E Stains Allows Granular Stratification of Oropharyngeal Cancer Patients. So Sebastian, thank you again for joining us today. It's really great to have you here. And when you're ready to start, please go for it.
So thank you very much for the kind introduction. I have to say that, given the authority of TIA and all the, superstars of lineup speakers, I rather feel, I would say, small in comparison. Nevertheless, I'm very thankful for having the opportunity to talk about this. I have to also confess that I haven't talked about this topic since a while because of different interests, right? But let's start with some biology and clinical relevance that we first focus on, and then take a look at the computational approach and the data, right? So as a first introduction, I think you would have to have an idea about what is going on with viruses in the human body. And probably it's not a big surprise to you that there are some viruses out there that have an oncogenic potential. And I'm almost certain that some of you have already heard about some of these. For instance, Epstein-Barr virus is very something that probably also is communicated in the public body. That's actually HCV, which causes hepatocellular carcinoma, which is less frequently in our localization of the Earth, but nevertheless. You are going to hear today about something else. You're going to hear today something about the HPV, which is a human papilloma virus. And human papilloma virus is among those family of oncogenic viruses, virus where the oncogenicity is relatively well understood. And we are going to take a look into some molecular foundations here. So HPV itself, it leads to the production of oncogenic proteins. These are called E6 and E7, and these have dedicated tasks. And I'm pretty sure that you already heard about some of these players that they are interacting with. So for instance, P53 is the protein that is expressed by the gene of TP53, and it's I'm pretty sure you're familiar with the fact that it's a master regulator of the mammalian cell cycle. So it's very conserved among several species. And what E6 does, it essentially degrades the P53, so the function as a tumor suppressor is not longer there. On the other hand, we have this molecule of E7, which is a little bit more difficult, but let's focus on one of this function. One of this function is interacting with a very prominent tumor suppressor molecule as well. It's called retinoblastoma protein in this case. Eventually, you have heard of the fact that there are some cancers within the human body where you have a dual loss of both of those molecules, actually, which is usually clinically very difficult to treat, including small cell lung cancer. But in this case, in the particularities of HPV, it leads to binding of RB, which leads to the release of the factor E2F, which has several effects on the cell cycle. Essentially, it's leading to cell cycle progression and therefore proliferation. And what is also important, it's that spike mechanism. It also leads to upregulation of a certain protein, which is called P16. So if you draw a bottom line under the effects of HPV, you can say it has a prominent role on the cell cycle and in tumor suppressor genes, and therefore is oncogenic. And if we take a look at the anatomical localizations of the head and neck area, you will realize that there are several regions where HPV actually plays a role. But you should pay a careful attention to the fact that usually we divide HPV into low-risk HPV and high-risk HPV, and these have distinct etiologies that they are associated with. So for instance, in the nasal cavity, there are certain papillomas that are associated usually with the low-risk HPV type, as well as some cancers that are associated with the high-risk subtype. And talking about the high-risk subtype and the topic that we have today is the oropharynx, where we have certain high-risk types, the most prominent one being HPV-16, but also 18 and others, which is associated to squamous cell cancer. And talking about the oropharynx, there's a catch that you have to consider when you talk about, as usual, diseases where certain subgroups can be identified, that there's a certain anatomy and certain regions are only included into the oropharynx. That is the tonsil, the uvula, the soft palate, and the base of tongue, areas that I have highlighted over here in the schematic. And the reason why I'm mentioning this is that there's a dedicated risk that is associated with HPV infection with this specific anatomical localization. And this is oropharyngeal squamous cell cancer, OPSCC. And you can see it's a disease with, this is data driven from UK and US databases. And you can see that the dashed line is essentially going up. So there's an increase in the incidence of oropharyngeal squamous cell cancer, especially this one also associated with HPV. And you should consider that is a disease that is usually found in younger patients. Younger patients without many risk factors. So it's not like they have smoked a lot of cigarettes or drank a certain type of alcohol. These are usually young patients that present with lateral mass, which is usually a lymph node. So they usually actually also present with metastatic disease to the lymph node itself. And the bottom line is it's a disease that has overall a good prognosis, right? But really, I want to make the point, and I'm going to go to the slide back just to make sure that I covered this, very well. So we are only talking about the subtypes of HPV positive and HPV negative disease within oropharyngeal squamous cell cancer. So it doesn't really make sense to divide those subpopulations within the larynx or the nasal cavity, because there is not this distinct survival curves as is occurring within oropharyngeal squamous cell cancer, right? So when you look into TCGA database, for instance, where you have a number of different anatomical localizations, it doesn't really make a lot of sense to divide these into HPV negative and positive and say, well, these ones have a distinct survival, because it's not entirely true, right? So the sub-definition of those subtypes really is important within the anatomical localization of the oropharynx. Talking about the disease itself histologically, is a disease that is defined by, as we can see over here, neoplastic epithelial cells, usually without keratinization. It's sometimes, but not always, accompanied with inflammation. And this I've copied this table. I've copied from a paper that we published 2021, where we built, I would say, the first model to detect HPV positivity from H&E slides. And just to show you that also pathologists as human observers can actually identify HPV infection within oropharyngeal squamous cell cancer. So there are certain features that humans can also see within those images. Overall, the metrics are not dramatically good, so the overall overlap between the observers is not great, but nevertheless, they are able to identify oropharyngeal squamous cell cancer. Talking about the subtype of the disease, namely the HPV positive and HPV negative OPSCC. There are guidelines how this is defined, and I've already told you, which I'm going to repeat in the next slide, but P16 is one of the surrogate markers that is used to essentially declare HPV positivity. Again, here is the slide that I showed you, where you can see that the release of this A2F transcription factor leads to upregulation of P16, which is then used diagnostically to determine HPV association. But I want to make the point within the guidelines over here, which is important throughout the talk, that actually what is required by the guidelines is P16 IHC, and there's an R for DNA HPV testing. So there are different methods how you can diagnose HPV positivity. You can run a certain assay which detects all the different subtypes. There are also more specific assay, but overall the guidelines say do P16 and eventually, in addition, HPV DNA testing. What it doesn't say, and I'm going to talk about this with the next slides, what it doesn't say is do both. It doesn't say you have to do a P16 IHC and a HPV DNA testing. So it's really like the guidelines say P16 is sufficient. And let's take this with a grain of salt. The author of the guidelines are very well aware of the literature, I believe. Probably it's still a little bit too early to make this a must-have. But it has been shown since a couple of years that if you imagine you have two markers, P16 and HPV, you can have 4 combinations. And if you look here at the red curve, so if you take a look at the red curve over here, you can see this is really the ones with a very good prognosis, which is usually defined as the hallmark of the HPV associated OPSCC with a good prognosis. You can see that P16 HPV DNA positive cases really do well. They usually have, in 5-year overall survival rate, about 80% or something. And if you take a look at the purple, which I think is purple, the purple line over here, you can see that these are the ones, so the HPV negative OPSCC cases that do worse. And the mixture of the two, which is not so frequent, but still, and nevertheless, they occur, they are in between those two. And this is something I believe, which is quite interesting, right? And there is, on the topic of HPV or OPSCC heterogeneity, there's an additional layer of complexity, and that is that you do not only have the heterogeneity because it's 2 different diseases, HPV positive and HPV negative with an OPSCC, but also HPV positive cases itself do have distinct clinical causes. And this paper by Crystal Lancues actually makes this point rather nicely, because what they showed is that by looking into HPV high risk, HPV subtype 16 positive cases, oropharyngeal squamous cell cancer cases, you will be able to define subtypes based on a certain certain mutations within the virus. And they could show that if you have an accumulation of those high-risk mutations, the prognosis of the formerly good prognostic HPV-positive cases is actually worse as compared to, I would say, wild-type HPV-16 positive cases. So talking about OPSCC, we already understood, well, there's a good prognosis associated with HPV. I just told you that there's heterogeneity on the level of HPV as well. And if we take a look at another paper at a different level, so this is not on the level on DNA, but it's on an epigenetic level and has been relatively recently. Maybe you can see the citation not so much because I'm in front of it, but it has been published by Sid Purham a while, not so long ago. The fact that by using single cell analysis of OPSCC cases, he could show that essentially what occurs is there's an epigenetic switch within HPV positive cases. So imagine you have 100% of the tumor with certain areas, and within certain areas, apparently, there are tumor cells where the clones shut down the expression of HPV DNA. And what he tells and what he defines as HPV on and on, HPV off phenotype, right? And he can rather nicely show in his paper that the HPV on and HPV off patients have a distinct clinical cause. So to me, another layer of complexity, just talking about HPV and the positivity, right? And I'm going to quickly sum this up because there was some biology, some molecular biology within it. So essentially, what we're dealing with is OPSCC is a disease that is with a rising incidence in Western countries. I should mention and strengthen the point that yes, there's vaccination that is possible for HPV. This has been shown very nicely for cervical cancer. I'm pretty sure most of you have heard about it. For oropharyngeal squamous cell cancer, people expect an effect in the next decades. So this is still a long time ago since we see benefits from the vaccination programs. OPSCC, as I told you, is rather in young patients. And I should also mention, so this is not my particular expertise, but I should also mention that the different treatment modalities that these patients have to go through are rather demanding. So you would have to imagine these are young patients with no major risk factors, and they have to receive a lot of therapy for the disease, right? And although we know that a good proportion of these will have a good clinical course, at least in Germany, the patients will be treated in the same way. This differs, of course, internationally within the guidelines, but nevertheless, we still have the challenge that we have a disease with a relatively good clinical course, but it's really hard to tell who's going to benefit from relieving the stress of the chemotherapy and radiotherapy for these individual patients. And yes, another point I made, I think this is pretty clear now, is that P16 can be used as a biomarker, right? So this is a surrogate marker of HPV infection. It has been probably 150 years ago since I approached Jens-Peter Klussmann. No, it was, I think, 2018 or 19 or something, when I approached him and said, well, look, there's this deep learning. We should use it to determine HPV from histological slides. I have a computer. Let's do it. And he was very supportive, and this is essentially how we shipped our first paper on this topic. What feels like really a long time ago. And I should also mention that this paper, I was sitting in the audience when Crystal presented this. Also what it feels like a long time ago. And I was really thinking after the session, I presented the first paper and she was giving the talk about this. And I was thinking, I have to do something with heterogeneity. I think this is the catch. Initially, I was thinking we should employ several strategies to just increase the accuracy, increase the specificity and sensitivity of the models. But after seeing this, I was thinking, well, I do not have the ground truth of all the different layers of complexity. I do not have the, I would say, high risk HPV subtypes. So if you can follow along my line of thought, I was thinking it is going to be hard without the real ground truth to make this the next blockbuster story. So I was thinking really, I probably should switch to some sort of heterogeneity within the disease. And well, I guess showing this slide for the audience here was an expert on these different niches and topics is probably, I know I don't want to tease you, but of course you always have to remember the fact that we're talking, when we're talking about whole slide images, we're talking about a matrix, right, of information. And what I've done is in the first place, I've been thinking, how could I eventually make use of some of the, what I was thinking was variability also within the slides. I just frankly used something as the variance itself, right? So I extracted the variance of the tie-class probability just because I was thinking, I know for sure that this is something, if I have a classifier that is more or less accurate to predict HPV presence, it's going to tell me how much variability is within a given slide. So this is really my line of thought in the very initial phase, right? And to give you the details, and what we put together then was a cohort, right? So as usual, within digital pathology, that is the most challenging task to get well annotated slides. And we were fortunate enough to have partners from Gießen, Cologne, and Maastricht, Heidelberg, and some cases we use from TCGA as well to contribute to this. And I think what is really unique and cool about this, that we have more than 90% of these have dual testing of HPV DNA, high risk testing, as well as P16 IRC. So I hope that I shipped you a lot of evidence that you believe that this would be a rather good idea, but of course you can also criticize this and say, I don't need this one. Fine. Well, we were very, very fortunate and happy that we had both of them. And I also want to make the point that within those, within those cohorts that do not originate from studies, there's always heterogeneity within them, right? Especially if you have different nationalities that have different treatment regimens, sorry, like Netherlands and Germany and CI, United States, you have to always be careful there. But nevertheless, mainly we have the classical surgery, chemotherapy, and radiation schemes that are applied within different centers, right? And this is a slide that's going to be very busy, but I wanted to just show you that essentially you will build these different cohorts, as everybody does. In this case, for the training and validation cohort. So I've decided to only use Gießen, TCGA, and Cologne for training purpose, because I was thinking it would be really cool to have one center completely left out of the training cohort, because I wanted to really see what the performance would be like for the model to have a complete different slide set, right? So we started with about 267 cases, and 267 cases. So my setup is a little bit difficult, and I'm trying to now, I have to do it this way. Within the test set, we were fortunate enough to have 639 cases. And what I think is cool to have is that we not only have these primary lesions within the test set, but we also have this metastatic disease, right? So I already told you that it's very common for the disease to have a manifestation at lymph nodes, at the head and neck lymph nodes. And the system itself was not trained on lymph node slides. So we have a test that is from different centers. It's from Gießen, Cologne, Maastricht, and Heidelberg. And that's essentially our way how we started. And to give you an idea about the computational approach, so I should mention that I haven't looked at the code since a while because of several other obligations, but I've written this. So what I want to tell you with this is I've written this a long time ago, right? And I'm not sure about you guys, but whenever I take a look at the code a long time ago, I can scream because there's so many things I would do differently. And this is, of course, true with this as well. But again, time is limited. And somebody told me that the most important thing you have is time. So I'm trying to be careful with my resources over there. But nevertheless, I'm going to talk you through the overall holistic concept of the computational approach. So the algorithm itself works as an input using whole slide images. So you can just have a directory with any kinds of, hopefully any kinds, so far, at least what I've tested, any kinds of whole slide images. And it will serve you with tabular data, with what I call high resolution thumbnails. So these kind of images that really show the whole slide with the prediction maps, as well as a QPath project, so that you are able to zoom and take a look into the details of the predictions. How this works is it's building a QPath project. I'm using Paku, which I found really handy coming in to do that. What I'm doing then is actually, which has some flaws, actually using the QPath inherent segmentation for just the tissue detection to then just write the overall tissue tiles within directory, where I'm then going to run an segmentation on the tumor area. After that, I'm going to extract those binary masks again into QPath, export the tumor tiles right into the directory. I'm going to apply a stain normalization, which I did, I think, using multi-threading. And then there's the classification step for HPV association. And the last step is going to evolve to visualize the results. So I'm using QPath for doing these thumbnails. and saving the tabular data, I think, in CSV. And together you end up with a folder essentially with all the information that you hopefully are going to have to need to rebuild the things that we did in the paper. And what I was thinking in the face of running the algorithm after I've essentially built all the different components, how I could really make use of the variability, right? So I was really intrigued by the fact, and I'm sure there are many ways how you could take a look at it, but sometimes especially for me, as I'm used to also take a look at the raw data and how they unfold, I was thinking there has to be something there, right? So what I've done is I've essentially thought, well, why not using the variance first as a kind of as a way to subdivide the set of the cases and see how our prediction of the overall survival will unfold after we filter for a certain variance. Because I was thinking we should get loose of this. If you remember, I can go back to the slide. This can be jumpy, and I apologize in advance. Yes. So if you remember this one, so I've been really thinking eventually we can, get rid of this ones, right? So I was thinking, okay, if we can sort out the cases where there is so much fluctuation, eventually we can really have the extremes that we are going to compare, right? So now I have to make sure to hit the right slide deck. And apologize if it's too jumpy. Here we are. So that was my thinking, right? And you cannot, I mean, there are many ways how you could do it. One way would be to, use the whole set. And I was thinking, well, then all the cases are burned because I, made an artificial threshold. What I did was I subsampled within the population of the test set. So these are 36 cases where I did a random subsampling of these 3 times. And I was thinking, I'm just going to take a look into, seeing how if I set it to certain thresholds with certain variants, how the different metrics are going to unfold. So with specificities, sensitivity, and the area under the receiver operator curve, right? So, and there was a certain threshold where I was thinking, okay, these are numbers look rather well, and this is my essential approach to, well, use of first of all the variance as a threshold and see whether I could stratify those patients more accurately. And this is in figure what I've just told you, right? So this is what I've done. I've done the filtering of this population, and by using this filter, of course, there's a fraction of patients that will not meet this criteria because they have a higher variability. And this is about 50%. So we are dealing here now with about 50% of the study population. You can see that I haven't told you the initial ROC curves, but you can see a slight increase in the performance of the algorithm to declare HPV positivity from 0.83 to, I think, 0.88. But what I was really thinking, what I was thinking was the catch was actually the survival data, right? So this was kind of cool, but I have just told you that I don't think it is really the overall goal to make the best HPV prediction algorithm because of the many flaws we have with HPV itself, heterogeneity. And so taking a look at the prognostic relevance, I was really intrigued. So this graph is something where I believe you can see that the biomarker has a lot of prognostic relevance, right? So we're looking here into the title's prevalence of HPV positivity. It's here called HPV association, but it's the same thing. You can see that this can be from 0 to 100. So you can have 100% of the tiles being positive or 0. And you can see there's a hazard ratio being plotted. And you will see an association with a score, which is to some degree almost linear, where you can see an increase within the score. Sorry, with an increase on the amount of tiles being HPV positive, the prognosis is getting better, right? And I thought this was really cool, right? So you have essentially a way to visualize that what many numbers can tell you in addition, but these are arrow bars. What you can see here within this Cox proportional hazard model that there's to me almost linear correlation, at least to a degree, with the score and the prognosis, right? And this is also highlighted by this Kaplan-Meier curve, which doesn't use no threshold like this one, but uses a 3-tier threshold with, I think, 20% and 80% and in between. And you can see that patients with a high score, so meaning that they have a high number of tiles being associated to HPV positivity, that they have a prognosis that is really good, right? So they have a 5-year survival rate of around over 90%, right? And so I was thinking, well, this is cool, right? So we have apparently a way to filter the cases. Cool, we can increase the algorithm performance, okay. But this was really cool, I was thinking, because we have now really a strong marker for prognosis. And let's go back to our cartoon. And well, I was staring at this, not actually, I was not staring at this. I prepared this for the reviewers because they wouldn't get my points. But anyway, so you can see hang on one second, you cannot see I'm going to put this here. Yeah, so I was really thinking, okay, well, I have to do something to combine this as a whole. And for whatever reason, I was really always intrigued of feature generation, by using just not ChatGPT, but my creativity. And I was thinking, well, I know from several projects that you can normalize the data very well using logarithm, and why not using just the variability itself times the class prevalence to essentially come up with a score that's going to reflect both. So we're going to have on one hand the tile class prevalence, which I just showed you has a nice correlation to prognosis, and on the other hand the variability. And the reason, I think this is super simple. This is just the variance. So I come up with this score, right? So and what I'm then going to show you is essentially the application of this score within 2 different scenarios. And if you are interested in the paper, there are many other scenarios, but I'm going to highlight 2. And the first thing that I did was, because I know that what I told you is that early stage OPSCC cancer patients, these would probably and most likely be the ones that could qualify for treatment de-escalation, right? So young patients, not a lot of risk factors. Eventually, if we can find, patients with a favorable prognosis within this population, it would be kind of cool. So running this for stage 1 and stage 2 disease, you can see the same thing. So I should probably take more time to explain this graph. So essentially what it is it's the error bars of the curve. And then you will see here the different risk, different hazard ratios that are associated with a given combined score. And you will see here the distribution of the values. Anyway, so what you can see here is that there's, again, a good correlation with what is now called combined score, essentially a combination of the variance and the tile class prevalence. So I would think a marker of eventually HPV heterogeneity. You can see that there's a nice correlation with survival. And what you can also see is that we can really identify a subpopulation of patients with a very good prognosis. And if you're interested in all the comparisons with regular testing, I can tell you that these regular 80% 5-year survival rate is also true for this dual testing within this early stage cancer, early stage OPSCC cases. We are around 0.9 something within the by using the computational approach. And this is also true actually in HPV positive cases. So I'm going to compare this in the second slide with something where I draw, in my mind at least, draw the lines where I think this comes from, although I have no evidence. Same thing, so we are going to take a look into our test set of a population which HPV positive disease. So this is only cases that have high risk HPV positive cases and P16 positive, right? 232 cases. Again, same thing, you can see a correlation with the combined score and prognosis. But what you will also see within the population of HPV positive cases, that there are subgroups, right? So patients that do well and patients that do not so well. And take a look at the numbers. So these are about 8 years survival rate, median overall survival rate for the population of what I would consider HPV negative, or in this case, combined score low cases. And please, again, take a look at the graph of crystalline Qs. And what you can see, again, I have no evidence whether this is really associated to it, but you can see that we have, I would say, similar, to some degree, similar curves, right? So we have also these HPV positive cases. So very similar to our cases, also HPV positive OPSCC. And we see that these are divided into subpopulations using, in this case, DNA testing with their single nucleotide polymorphisms. And you can see that there are patients with a good prognosis and patients with a bad prognosis. And it's very intriguing to me that these lines, to some extent, look similar, right? And I can also tell you that, of course, the system is not perfect. I mean, not perfect in many ways, but also not perfect in terms of the classification, right? I just wanted to tell you that for some cases, for instance, these are cases where the algorithm has declared positivity. So the algorithm said, the algorithm said these are 2 cases that are HPV positive, or otherwise they are scoring with a very high combined score. Interestingly, they both are P16 positive. And if you take a look at the prognosis of the patients, I wouldn't say it is dramatically good, but it's also not bad, right? So 12 years and about 7 years for these 2 patients, the durations, how long they lived. So I was thinking, well, nevertheless, they have been classified incorrectly because they have been HPV DNA negative and P16 positive, but overall would be HPV negative. But nevertheless, I wanted just to tell you that I believe this was not really to the disadvantage of the patient, right? And the same thing is also true more dramatically for the ones that are positive but have been classified as negative. So these are HPV DNA positive and P16 positive. And you can see that they are, well, you have to take this with a grain of salt because they're almost dead until the time that they are monitored. But I think what you can appreciate is that the prognosis of these patients is not very good, right? Although they're HPV DNA and P16 positive. And the algorithm has assigned them really to the low category, to the HPV not associated to category, right? And this was pretty much my take on the story. And you see that the next steps are pretty blank. Because I was thinking, if I have now the opportunity to talk all with the brilliant minds from TIA, that I'm pretty sure that they know the way to go. So I would be interested to just hear your thoughts on the overall study and all these kind of things. And I'm pretty aware of some of the papers, at least, that came from TIA. I know that I remember HOVONET, which I believe came from TIA as well. Hopefully there's nodding. Yeah. I actually used this as well. I also remember a paper for OPSCC using HOVONET and the distribution of immune cells, which was very nice. So I'm very happy to take some questions or feedback.
Brilliant. Well, thank you very much, Sebastian, for a really great talk. So yeah, should we open the floor to questions then? Has anyone got any questions, comments, or start with? Anyone online or in person? So I'm going to start with a really broad question. So first of all, so that was a great presentation, really interesting. So how do you expect it to, do you expect this sort of algorithm to work well in terms of different types of head and neck cancers, laryngeal cancer or oral cancer? I know it's obviously oropharyngeal, but is it the case that HPV positive association is not necessarily as important, or do you expect it to work, I suppose?
Yeah, I think this is a good question. I'm going to put the slide again. I would only limit my expectations to OPSCC. So it is true that there are some diseases where the link to the development of the disease has been drawn to HPV. But let's take one example. So there has been a time where there has been a subtype of papillomas within the nasal cavity, and one was thought to be HPV positive and the other not. It was just an epiphenomenon. What we know from those anatomical localizations is that only HPV matters dramatically for the prognosis of these patients. So I think you could, of course, put an algorithm and analyze and see how this is going to predict HPV presence in some of these localizations. But my personal take would rather be to use it for the prognostic relevance, which is not important for laryngeal cancer, for instance.
Okay, thank you. That makes sense. And I know in one of your very early slides, you talked about the incidence of oropharyngeal cancer increasing quite a lot over the last 10, 20 years. Why is that, especially if across the world, is the number of sort of smokers increasing, or what's the rationale behind that?
Yeah, I'm going to see whether I can jump to the slides. So actually, is it this one? Yeah. So this review by Axel Lechner is having some good tables to get an idea of the risk factors also for OPSCC, and smoking and tobacco use is not one of them, actually. So there are two things. The incidence is rising primarily in Western countries with a high income. And the second thing is, why is that? So I think this had also something to do with the sexual lifestyle that has changed over the years. And there are many other factors that contribute, but I think this is something.
Okay, thank you. Do you have any more questions for anyone online or in person?
I guess when you were talking about the...
Sorry, can you hear that?
Mm-hmm.
Can you hear Mark? Okay, brilliant. Sorry, carry on, Mark.
You were talking about the graph of the hazard ratio and saying there was a linear-ish correspondence. Was that just because most of the cases were in a smaller region where it was roughly linear? Because the curve itself looked very much not linear. So I was just wondering what the thinking behind that was. Is it just that most of the cases are in a relatively small section of that graph, and within that small section it's roughly linear? Is that...
I probably said this because I'm used to explain this graph to people without a mathematical background, right? So you're completely right with what you're saying. I believe the right word would be correlation, right? So, and you're also right with pointing out that within the region where you could assume there is somewhat of a linear correlation, this is around 1, where this is, probably not the right wording.
Yeah,
So
but I guess, yeah.
Let me rephrase it to correlation. And the reason why I put correlation here is that whenever you talk about these kind of graphs, I think it is very difficult to really understand the prognostic relevance of a biomarker, because there's a threshold being set, right? And what we know from the metrics that we came up with in the paper is that the score is really associated to prognosis in a way that it's not an arbitrary threshold that's being set, right? So usually when you take a look at these graphs, you would have to curiously ask how it happens if you set the threshold into different limits, and how the graphs then look like. And I just tried to make this point by plotting these graphs which show this association.
Okay, makes sense. Thank you.
Thank you. And also sort of continue on that. So I think I understood the rationale behind using a three-tiered system that you've got there as an output. But did you try it using just high and low, for instance, and just having one single threshold? Did it not quite have the same results, or what was
Yeah, so absolutely. And I mean, two things. So depending on how you put the threshold, you can mimic that the prognosis is very similar to HPV prognosis. So the 80% 5-year survival rate. And I was thinking, okay, this is kind of making the point that we are detecting HPV association, but we would be interested in identifying a subpopulation within better prognosis. So the answer to the question is I've set 1 billion thresholds. At the end of the day, I was thinking this 3-tier threshold still has, well, it still has this fraction of patients with this excellent prognosis, where I think there's really something that can be used clinically. And at the same time, you can still run the statistical test and say it's, you know, it's relevant or it's irrelevant.
Okay, makes sense. Thank you. And I suppose because we're all, well, a lot of computer scientists in the room at the moment, so you know, I really like the clinical the pipeline that you showed. But have you got any more? Could you give us more details about the actual algorithms that you used in terms of segmentation and classification?
Absolutely. So let me try to find the slide.
And
yes. So if you take a look into the paper, the GitHub repository is associated with all the code that you would be required to run the algorithm, right? And I wasn't really sure whether the time would be enough to go into the details, but I can quickly mention some points. So as I already told you that as most computer scientists, I like to have at least a Python backbone. I haven't applied Rust yet to any of those projects, but eventually in the future. I'm just joking. Let's take a look at maybe, for instance, yeah, I mean, this was, so the segmentation is an FPN with a ResNet 18 encoder. I was thinking when I was designing all the different components that it would be also cool to have this rather lightweight. I tried to parallelize the processes within the several steps in order to make sure that it's fast. I believe it is fast. But it could be faster because there could be ways also to improve it. But nevertheless, so this is an FPN with a ResNet 18 encoder, and the stain normalization. So for the training purposes, I actually used Bakenko, Reinhardt, and the other one I always forget. But I used 3 different ones. When we're talking about the training phase, I tried to use an augmentation based on H&E variability by applying those stain normalization techniques. So these stain normalization techniques have different flavors, and I believe that more and more people get rid of them. At this phase, and again, I've written this a long time ago, I was still happy with it. And for running the classification, I'm still using the stain normalization step because I was worried about an effect on HPV prediction based on color variation that I couldn't really rule out with the 4 or 5 centers that I had access to. And the classification then runs using a ResNet 18. So I've tested several networks in terms of the performance, and I could see what is not completely surprising, that ResNets perform very well. ResNet 50 was comparable, but a little bit, was comparable to ResNet 18. But as I wanted a lightweight backbone, I decided for ResNet 18 actually. And the beauty also of these rather smaller networks is that they don't have this gigantic fingerprint of some of the variability that you find within the slides. So sometimes these more lightweight networks also have an advantage when it comes to generalizability, at least to my experience, right? And yeah, well, the other points really I try to put into QPath. I can quickly show you. I mean, this is probably for how is it called? Having a laugh, but I can just quickly show you what I recorded in high speed. So this is from the command line. So there's several, so I'm going to stop here. So because this is high speed, right? Within the repository, I have a command line and a Streamlit application. So during the time when I was thinking that people would use it, I was thinking eventually Streamlit could be an option because it's more user-friendly. And what you can see is here that it followed all the different steps automatically, ending with, here in this case now, 3 larger thumbnails with the overlay of the predictions. And yet you can see here the tabular data, and it actually also is fed with a threshold from the paper so that you would be able to declare the different scenarios for the given case. And at the end of the day here, I'm going to import the QPath project so that you're really able to zoom in the different details and have the classification maps. And I was thinking at the time that, QPath is rather handy, especially when it comes to zooming in and seeing how the mistakes or the corrected, the mistakes or even the areas where the segmentation could be improved. And this is pretty much the algorithm. And if there's anything specific in terms of the questions of the algorithm, I can kindly point to the repository, and I would really apologize because I do not mitigate errors there. As looking at it, I see many things I would do differently, but again, there's limited time. If there's anything, so I tested it on 2 or 3 machines, it worked. And you know what they're saying, it works on my machine. So yeah, be patient with me if you find anything funny there. But yeah.
That's fantastic. So the code's all available online then. That's brilliant. And did you say the data was available as well?
So the data, you mean the training data?
Yeah.
Yeah. So I haven't made them available because I was thinking at the time with illegal activities that I didn't want to go through. And so I'm not planning a particular future with OPSCC classification because of other responsibilities, but I'm pretty sure that you could eventually approach Jens-Peter Klussmann to see whether he would collaborate. Otherwise, I can also, I have really legally this is not dealt in. We would have to anonymize the cases probably and take it from there. So I do not have a direct answer to this. I was initially thinking of doing it, but I haven't done it. So I just released the weights and the network itself.
Great. So has anyone else online or in person got any further questions they'd like to ask? No? Okay, well, I'll finish with one final question then. So given your experience, what would you say was the next steps of the future, I suppose, for computational pathology over the next, or at least over the next 12 months?
Can you please repeat the question, because I wasn't really sure whether I understood the question, but I wasn't really sure whether I got the point.
Yeah, well, in your opinion, what do you think is the future of computational pathology in the coming year or so?
So it's a very funny question. So I have to be careful what I'm saying now. So I personally I realized by myself that I got toxified by reading the amount of papers that's being published to some extent, right? So there are really some people that have brilliant papers where I feel like this is the way to go. And then there's so much, I don't know what to call it. It's very diverse. At the same time, this is research and science. You always have, when there's a new technology, you always have this, you have this increase, which is also great. So I think the future is going to be very much coupled. So I think that given the increasing amount of data that these models are trained with, I think we are going to see less and less stain normalizations. So from my experience, they are important and they increase the metrics. But I think that once you get diverse data sets, they really, can be also exchanged. It would be cool to understand what you guys are thinking about it. So I'm going to go on and maybe you can comment on the points. Second, I think that this, so I always was careful by doing this end-to-end training. I'm a pathologist, and I always see the complexity of the different cases. And to give you one example, I don't need an algorithm to detect a microsatellite instability because I do this with the IHC within 10 seconds, right? And this is very accurate. And I was always thinking about, when I'm going to build an algorithm, is there really a niche, or is there really an application, despite having a paper to say you detected something? And I think what we're going to see is a broader application of these algorithms. If I take a look at the different large language models that are going to be coupled now with computational pathology, there's going to be much more interaction, right? And I'm not so sure whether we are going to see many of those papers that I've published in the future, where we have this essentially proof of concept scenarios. I think we're going to see more holistic models there, eventually an head and neck model or module, where you can ask specific questions like, is there a signature for HPV, something like this. So I think probably these things are going to be more complex. What I'm still hoping is that there's still some willingness to couple this also with the molecular understanding, and really to make sure that we have that we also excel on the molecular understanding of the diseases, right? So good IUC curves are great, right? And eventually this is also not the responsibility of the whole field, but I think we should also keep this in mind, that these are also fundamentally linked sometimes to biological processes. So the point I want to make with this is, if you discover that your model is not good at something, maybe there's something interesting, right? So maybe and this is really cool to have a diverse team, where somebody from the field can say, hey, look, I have an idea. Maybe this subtype that always stands out, and you find out something. So I think really take a look above your table there, and eventually be prepared to discover something.
Yeah, definitely. No, thank you very much for that interpretation, for your opinions in the future. It's I completely agree with the first point you said about stain normalization. I think in our lab we've generally stopped using stain normalization quite as much, and we tend to use standard augmentation to train the models, has been the sort of our approach for the last couple of years. I don't know if anyone else has got any comments they want to make about that at all, but and that tends to be.
Yeah, I agree. I'm mostly doing augmentation.
Yeah, and in terms of, I think your second point, in terms of foundation models, again, I think that's a really exciting aspect, I suppose. And if we could get some sort of foundation model for head and neck pathology, then I think that'd be amazing. I suppose collating the amount of data in the head and neck might be difficult, but yeah, I agree with that interpretation. I think again, if anyone else has got any comments to make on that, feel free to.
Yeah, I think another point on the, I guess the stain normalization stuff, as we get more and more of these well-performing sort of foundation models that have been trained on just a massive bunch of diverse slides, it becomes less and less important from that perspective as well. If you're building your algorithm based off of one of those foundation models, you then, again, don't have to worry so much about stain normalization. It's kind of agnostic to stain variation, just through being trained on such a wide variety of data. I think, yeah, as foundation models become more of a thing.
I'm brilliant. Thanks,
Sorry, that there's still comments, because one thing I really find also fascinating is a field that I haven't really started to excel on, is this whole topic of pruning. And after you build these foundation models, to really start making them lightweight and improving the performance there. I think there's a lot of things that need and can be done there.
Yeah, definitely. I mean, we haven't done too much work in our lab on foundation models, have we, in general? Has any, well, you've done
just from using that, I found I've been using C-transpass quite a bit recently. It seems to be pretty decent, and we've had some talks recently which have shown that when you use these foundation models, stain normalization has less of an effect. So there's actually been some results we've been seeing from a few other speakers that have kind of numerically backed up that intuition, as it were. Yeah.
Brilliant. Well, has anyone else got any further comments, questions, anything to finish with? Or should we call the seminar to a close there? Well, thank you very much, Sebastian, for a really great talk. Again, it's been really interesting speaking to you and really interesting to hear about your work. And no, best of luck with the future. And I'd just like to remind everyone again that the MIUA 2024 special session deadline for short abstracts is on Wednesday if anyone wants to submit an abstract. And we've got a few weeks off until our next one. Our next seminar will be on Monday, the 10th of June, from Dr. Drew Williamson from Emory University. So I hope to see you all there. But again, thank you very much, Sebastian, for a really great talk. And it was nice to meet you.
Thank you very much.